Okay, we'll be looking at the last part of part 1 of the extension topic A on the immunological memory. Alright, so just a recap on the specificity and the memory, which are the hallmarks of the adaptive immune system. So for this video, we'll be focusing on the memory, which refers to the ability for a larger magnitude and a faster, stronger response to the same pathogen for subsequent encounters of this same pathogen. All right, so looking at the difference between the innate and the adaptive immune system. For the innate immune system, as you can see here, right, each time the innate immune system encounters the pathogen, whether it's the same pathogen or a different pathogen, each time is the same, right? There is no increase in magnitude or the speed of response, which is represented by the diagram here, right? The speed of response, the time needed, right, for the innate immune system to respond is pretty short, because the innate immune system is always ready. It is the first line of defense, right? As for the magnitude, which is represented by the size of the figure here, right, it's the same each time, even though these are subsequent exposures to the same pathogen. But for the adaptive immune system, that is different, right? Adaptive immune system takes a longer time, represented here, to be triggered, but subsequently, for the same exposure to the same pathogen, you can see that the time needed for triggering of the adaptive immune system is shorter, meaning it is triggered faster, and the magnitude given by the size of the figure, it is larger each time that the adaptive immune system encounters the same pathogen. Right, so that is defined as the immunological memory displayed by the adaptive immune system. All right, so that is mainly due to the memory cells. And for memory cells, in the previous lecture, right, we have focused only on the memory B cells, because that's what the syllabus seems to focus on. But you should know that memory cells, that there are also memory T cells, be it the T helper cells, be it the cytotoxic T cells. They also form memory cells as well, right? As well as the B cells. Okay, so this part of the lecture, I think I'll just call them memory cells in general. All right, but you know that that it would include the memory T and memory B cells. All right, so anyway, the memory cells are produced when the adaptive immune system responds to the pathogen for the first time. Right, so we call how the naive cells are activated, and after activation they undergo clonal expansion. Right, so in the pool of the cells, be it T cells or B cells, some would differentiate into memory cells, which would persist in the body, right, even after the pathogen has been cleared during the first encounter or at the end of the first encounter. All right, so these memory cells would confer immunity to this pathogen. So when this pathogen reappears, when it invades our body again, the memory cells are able to very quickly respond to these pathogens much faster than the naive cells, and the response that they give would be also stronger and of a larger magnitude than the naive cells. All right, so for memory cells, a couple of defining properties. Okay, they are able to survive, they are able to persist in the body, but in a rather quiet or quiescent state. Okay, so they do self-renew, right, because they persist, right, in the body, but they do so via very slow proliferation, so they do not divide and keep dividing very fast, right? They are able to divide slowly, right? They have a longer lifespan, so they can then divide not so frequently to renew themselves. All right, and they are able to provide or give a larger response to the same antigens than another naive cell recognizing the same antigen. All right, because the number of memory cells for the particular antigen will be larger than the number of naive cells. So remember that, let's take B cells as an example, yeah? So you have different naive B cells with different B cells receptors that recognize different epitopes, right? This is what we have discussed in the previous lecture. All right, so for the same antigen, one antigen may have, say, a few epitopes, right? So a few of the naive B cells will be able to recognize the same antigen because by recognizing different epitopes, right, on the same antigen. But that is one naive B cell to one epitope, right? So for one antigen, maybe a few naive B cells. But remember that after the naive B cells are activated and have undergone clonal expansion, some of the some of these B cells, right, would go on to differentiate into memory B cells, right? So which means for each epitope, there would be more than one memory B cells that recognize this particular epitope, right? So for the antigen with the different epitopes, then there would be a larger number of memory B cells that recognize the same antigen than the naive B cells. All right, so I hope this explanation is clearer. All right, so there are more memory B cells recognizing the same antigen than the naive B cells. Therefore, when they respond, there will be a larger response to the antigen and to the pathogen than the first time, right, by the activation of the naive cells. Okay, and the memory cells are also able to respond more quickly. Okay, so you have this graph in your notes, right? So this is just some more description of the differences between the first exposure and repeated exposure. Right, so I'm not gonna talk too much about this. I think I'll leave you to read these descriptions yourself, which explain the difference in the shape between the two curves, right? For the first exposure versus the repeated exposures. Okay. Right, then let's move on to the primary and secondary immune responses. The primary response refer to the first encounter of the pathogen, and secondary responses would be the subsequent encounters. All right, so in the primary immune response, we can see that the levels of plasma cells and activated T cells, be it helper or cytotoxic, for the pathogen, right, would increase slowly, and the memory cells will be produced at the end of the primary immune response, right? Then they will persist, right, in the body. And when the pathogen is encountered again, right, the pathogen with the same antigen is encountered again, then the memory cells that were produced in the primary immune response will be able to respond very quickly. They will be able to differentiate into the effector cells. So for the memory B cells, there will be the plasma cells. For the memory T cells, there will be both the helper and cytotoxic T cells. Okay, and this differentiation, this activation does not require the antigen presentation by the antigen presenting cells, nor is there a need for cytokines from activated helper T cells. So that explains why the activation and the secondary response for subsequent encounters of the same pathogen would be faster, right? There is no need for the interplay or the action of the other cells. the memory cells can straight away differentiate into the effector cells, straight away can produce antibodies, straight away can kill infected cells without the need for antigen presentation or cytokines, right, from helper T cells. All right, so you have the same graph, right, in your notes. Right? I'm just gonna look at the graph bit by bit, right, in a more detailed manner. So hopefully you can understand the primary and secondary responses more in depth. All right, so for the primary response, right, that is when the pathogen and the antigen on the pathogen is encountered for the first time. Right, so focusing on the B cell, that would be when the naive B cell that has a specific B cell receptor would bind to this antigen found on the pathogen. Okay, so the naive B cell is activated, of course, aided by the cytokines from the activated helper T cells as well. Right, so that would then bring about the clonal expansion. All right. So here, right, would show you a diagram on how the activation of the naive B cell occurs, right, with both the binding of the antigen on the pathogen to the B cell receptor, which is a membrane-bound antibody, as well as cytokines from a activated helper T cell. Right, so this one we have discussed earlier, right, in a previous lecture. Okay, so here just a reminder. Right, let's move on. Right, so again, the activated B cell would first divide, right, clonal expansion, and then in this pool of B cells with the same specificity for the same antigen on the same pathogen, most of them would differentiate into plasma cells, and some would differentiate into memory B cells. Okay, so this explains why in the primary immune response, when the immune system encounters the pathogen for the first time, there is a lag before the antibodies can be produced, because time is needed for activation of the naive B cell, for clonal expansion, and for differentiation into plasma cells in order to produce the antibodies specific for this antigen on the pathogen. Okay, so that is why for the primary immune response, antibodies are only produced in this graph about 10 days later in this graph. Okay, so normally it will take a few days, right, for the antibodies to be produced. Okay. Right, so the antibodies produced in the beginning, right, would be mostly the IgM, unless or until class switching has occurred. All right, so in the beginning, right, primary immune response mostly would be IgM. There may be small amounts of IgG or even other classes of antibodies if class switching has occurred. So that again explains why there's a lag for production of antibodies, because class switching has to occur, especially for antibodies of other classes, IgG, IgE, IgA. Right, so at first mostly IgM. Right, so as the antibodies have been produced, as the antibodies carry out their functions, that aids the removal of the pathogen. Right, so at the end of the infection, most of the plasma cells would die via apoptosis, but the memory cells, the memory B cells here, as we are focused on the B cells, right, the memory B cells would persist in the body, and they would be specific to the same antigen of this pathogen that has been encountered for the first time. All right, so that brings us to the secondary immune response, when the body encounters the same pathogen with the same antigen for a second time or subsequent encounters. Okay, right, so the memory B cells, again, we are focused on the B cells here, so do not forget about the T cells as well, but we are focused on the B cells in this graph. Okay, so memory B cells would be able to respond very quickly to the same antigen. They would bind to the same antigen and also undergo clonal expansion and differentiation into plasma cells, but these does not require the helper T cells or the cytokines from the helper T cells. Okay, so that's why the response, right, and the differentiation into the plasma cells is much quicker, and to produce antibodies much quicker. So in this graph, right, it takes about 5 days, which is about half the time of the primary immune response given in this graph. Okay, just this graph alone, yeah. So normally it will still take maybe 1 to 2 days, right? But it will be much quicker than the primary immune response. All right. So as I mentioned already, right, the memory B cells would be able to recognize the antigen very quickly, undergo clonal expansion, and differentiate into plasma cells very quickly, much quicker than the primary immune response. Okay, so antibodies will be produced, and here the amount of antibodies will be higher, as you can see, than the primary immune response because there are more of the plasma cells, right? Remember that some we mentioned there are more, okay, even though this graph only show 1, but there are more of the memory B cells that would be able to recognize the same antigen because at the end of the primary immune response, the memory B cells, there will be not just one, right? There will be a pool of them. They would persist, right? So there will be a pool, multiple of these memory B cells. So when they undergo clonal expansion to form plasma cells, there will be more plasma cells formed than the clonal expansion of just one naive B cell in the primary immune response. Right, so there are more plasma cells that are going to be formed in the secondary immune response, therefore giving a larger amount of antibodies, and the antibodies would be mostly IgG or IgA and IgE because class switching would have already occurred in the primary immune response. Okay, so instead of IgM, the plasma cells that are formed from the memory B cells would be able to produce a different class of antibodies already. Okay, so that would be the secondary immune response, and of course after that the plasma cells would still die at the end of the infection, and more of the memory B cells, because not all the memory B cells would form plasma cells, right? Some of them would remain as memory B cells. So these memory B cells would still persist in the body for a subsequent encounter of the same pathogen with the same antigen. Okay, so lastly, right, just a table which you have in your notes as well, showing some differences, right, between the primary and secondary immune responses. All right, so I'll just leave you to read this yourself. Yeah, okay. So that's the end of part 1 of infectious diseases.